Okay. So, I'm going to continue on where I left off with regulating blood pressure. I added a few extra slides that I didn't have previously on here. I don't have them to give to you because I've picked them up from a previous textbook, and I don't quite know if I'm allowed to let you have, rights to download those or not. So I'm just not going to run the risk here. So we started talking about in class about the different reflexes for regulating blood pressure. And we started to go through this here, which I still think is important. And if you can understand what's happening in this flow chart, you will be well on your way. In fact, if you can write it out and kind of recreate it, it'll just help you to memorize and learn what's happening. But I'm going to give you some graphics that I always visualize when I'm thinking about the neural and endocrine regulation mechanisms associated with blood pressure regulation. So again, these are already in your slides, so you can see the different things that are affected. The baroreflexes, remember, barometric changes or changes in pressure or stretch that's detected primarily or detected by the blood vessels, and the neural baroreceptors are going to be associated with the aortic body and the carotid body, and so that's what this here. You can also see that mechanoreceptors are involved as well. So your receptor nerves, which is typically cranial nerve 9 and cranial nerve 10, vagus and glossopharyngeal, are going to be detecting what's happening at the level of the carotid and the aortic bodies. They're going to send that information to the medulla oblongata, which is going to bring it to the cardioregulatory center, and then that will in turn tell the body what to do to the blood vessels and to the heart. So let's say that the receptor detects an increase in blood pressure. That means you're getting increased stretching or increased baroreceptor firing. And what that's going to do is it's going to increase the cardiac inhibitor center, right? So think about that. So it's increasing the inhibitor. The inhibitor is what's going to lower blood pressure. So if I increase inhibition, I'm going to slow heart function down in order to bring my heart rate, my stroke volume, all the things to lower blood pressure. Think about it as a feedback loop. High blood pressure says we need to activate the mechanisms to lower blood pressure. Low blood pressure says we need to activate the mechanisms to increase blood pressure. Okay? So with increased stretching, you're going to see an increase in the cardiac inhibitor center, a decrease in the cardiac accelerator center, and then this is all part of this cardio regulatory center. Okay, so you have inhibitors and accelerators. The inhibitors, think parasympathetic, rest and digest. Accelerator, think sympathetic, fight or flight. And then we're going to also decrease vasomotor center, which is associated also with the sympathetics. Okay, when we increase inhibitor, decrease accelerator, and decrease the vasomotor center, we're going to see a decrease in cardiac output and an increase in vasodilation. Both those things lead to a drop in blood pressure. So decreased cardiac output, decreased peripheral resistance by increasing vasodilation will cause blood pressure to drop. And then we're back to normal, hopefully. When the blood pressure is too low, we see a decreased stretch and a decreased firing of the baroreceptor. And we're going to see the opposite of what we saw in response to the increased blood pressure. You're going to see a decrease in the inhibition, an increase in the acceleration, and an increase in the vasomotor centers. That will cause an increase in cardiac output. It'll also cause an increase in vasoconstriction. Remember, vasoconstriction is a function of peripheral resistance. So when I increase peripheral resistance and I increase cardiac output, we're going to see an increase in blood pressure. Okay, and so that's how the baroreflex works. For the chemoreflex, it's just about monitoring chemistry. Okay, so the receptors, again, are the chemoreceptors. They're located in the aortic arch. They're located in the subclavian arteries, the external carotid arteries. All of these are looking at chemical changes. And what are they looking at? They're looking at pH, and they're looking at oxygen and CO2. Now, you actually have two types of chemoreceptors. You have peripheral chemoreceptors, which is what are listed here, the carotid arteries, subclavian, aortic arch. And then you have central chemoreceptors. The central chemoreceptors are actually located along the medulla oblongata and the brain stem, and they are primarily detecting CO2. So your central chemoreceptors are going to be monitoring CO2. Your peripheral chemoreceptors are going to be monitoring oxygen. pH is actually going to be monitored by the central chemoreceptors as well, because the CO2, and we'll talk more about this when we get to the respiratory chapter, but CO2 influences pH, meaning when you have more CO2, you have a lower pH. When you have less CO2, you have more pH or higher pH, and we'll talk about that relationship. So again, and I started mentioning this, again, we're talking about the neural reflexes. So we're going to see when the cardiac centers and the vasomotor centers are triggered by, let's just say low oxygen is the main thing, then we're going to see vasoconstriction, and we're going to see vasodilation depending on the tissues that need to be vasoconstricted or vasodilated. So we're going to see affecting the heart, the lungs, and the blood vessels based off of the local responses and the automatic responses that are occurring in response to the concentrations of oxygen and CO2 and pH. All right, I'm going to get into these in just a moment, and we'll talk about these regulators, but again, just following the blood chart. So these are the graphics I got from McGraw-Hill, which is another textbook company that produces the Anatomy and Physiology textbooks. We've had a lot over the years, and I used these figures a long time ago, and they're still applicable. So this right here just shows an overall graphic of where the vasomotor center or the cardioregulatory center, they're all pretty much the same, located within the same region of the medulla oblongata. And when we're talking about the vasomotor center, it's what's controlling the blood vessels. So the only thing that controls or sends signals to the blood vessels are your sympathetics. Okay? Your sympathetic nerve fibers are going to cause vasoconstriction when they're activated, and when they're not activated, the blood vessels relax, and then they will start to vasodilate. And then what controls how much they vasodilate is if nitric oxide is released, if there's acetylcholine being released, those things, but they're not under parasympathetic control when they're vasodilating. It's the sympathetic nerve fibers that drive vasoconstriction when they're activated, and when they're not activated, they're turned off, then the blood vessels are able to relax. This is just another way to visualize the baroreceptor reflex. So here's our heart. Here's the aortic arch baroreceptors, or the aortic body. Here's my carotid sinus, which, it would actually be, notice that this is showing it in the brachiocephalic trunk, but it would actually be up into the carotid body. And notice that you have two nerves that are monitoring these things. You have the vagus nerve associated with the aortic body, and then you have your glossopharyngeal nerve associated with the carotid body. Now, this is actually interesting. When someone is having a heart attack, sometimes they'll complain that they can feel or taste a bitterness in the back of the throat, and that's because the glossopharyngeal nerve, cranial nerve 9, is responsible for sensory information dealing with the taste of bitterness in the posterior 2/3 of the mouth. So not only is it associated with the heart, but glossopharyngeal tells you tongue and throat, and it has to do with bitterness. And so that's just something that, another little side note, just in case you hadn't heard that one before. Okay, so they're sensing information from the heart. The vagus nerve senses information from the heart, but the vagus nerve also has motor activity, and it sends parasympathetic fibers back to the heart. And it will respond when, since we're talking about baroreceptor reflex, so it will respond when the blood pressure is too high. When the blood pressure is high, the vagus nerve is stimulated to cause the heart to slow down. And And then if the vagus and the glossopharyngeal nerve detect that the blood pressure is too low, then we're going to skip the parasympathetic aspect, and we're going to go down and activate the sympathetic chain ganglia. The sympathetic nerves will cause vasoconstriction, and the sympathetic nerves will also trigger the increase in cardiac output, so increase rate and strength of contraction done by your sympathetic nerve stimuli, and that has to do with the endocrine and the not endocrine, the epinephrine and norepinephrine being released in conjunction with the activity of the sympathetic nerve fibers. And in case you're wondering, this is actually known as the cardiac nerve fibers. Is what that is. So the sympathetic nerves that go to the heart are referred to as the cardiac nerve fibers. All right. Now, in addition to when we trigger the sympathetic nerves, one of the reasons why the sympathetics are on a chain ganglia is because we need to get, in order for the sympathetics to maximally function, they need to get out of the spinal cord, and then you get into the sympathetic chain. The sympathetic chain has nerve fibers that go directly to the adrenal medulla, and it's the adrenal medulla where we get epinephrine and norepinephrine. And a way to remember that is epinephrine is also known as adrenaline, so adrenal in, and then noradrenaline. Now, the difference between epinephrine and norepinephrine is that nor just tells you that a methyl group, a CH3 group, is just attached in a different location between the different molecules. Now, because it's attached in a different location, that's why you get different responses with epinephrine versus norepinephrine, but they're not opposites of each other. Technically, the opposite of epinephrine would be acetylcholine, because acetylcholine is typically associated with the parasympathetics, and epinephrine tends to be associated with the sympathetics. But you should also know that epinephrine, norepinephrine, and acetylcholine can be released by other factors and act as endocrine hormones as opposed to neurotransmitters. And so if you're wondering, is there a difference between endocrine hormone and neurotransmitter, in reality, there's not much of a difference. It's just a matter of how it was triggered or how it was stimulated. Let's look at the chemoreceptors. Notice that the same characters are involved. You have the glossopharyngeal nerve and the vagus nerve, both detecting the aortic and carotid bodies. They're sending the information to the cardioregulatory center. But what are the chemoreceptors looking for? Well, the peripheral chemoreceptors here are detecting oxygen levels. The heart, or your body, your brain, your body wants to know as early as possible if we're having trouble getting oxygen to the brain or to the rest of the body. And so we're monitoring oxygen levels at this level. We even have chemoreceptors in the kidney that are monitoring oxygen levels, as well as blood pressure. So there's baroreceptors down in the kidney as well, and we'll talk more about that when we get to the kidney. So the glossopharyngeal nerve, cranial nerve 9, vagus nerve, cranial nerve 10 send their respective information to the cardioregulatory center. Notice it's located in the same areas where we find the vasomotor center, and then what do we call the cardioregulatory center is probably a better description of everything. But you have, when we talked earlier about the cardiac inhibitor and the cardiac accelerator factors, all those things are going to be triggered and started off in the cardioregulatory center. So if oxygen levels are good, or maybe even slightly elevated, we might activate the parasympathetics in order to help regulate or maybe even slow down the heart rate. If oxygen levels are too low, though, we're going to go into a fight or flight response. But, and just because we say fight or flight response doesn't mean like you go into like panic mode. Your body just kind of switches in between, kind of like you're driving a car. So you hit the gas, and then you hit the brake, and then you hit the gas, and then you hit the brake, and you're just driving kind of like normally. Sympathetic nerves are your gas, and the parasympathetic nerves are your brake, and we basically drive with that system where we turn this on while we turn this off, and we turn this on while we turn this off, and just try and find a healthy balance in order to using that scenario to maintain the speed limit. So don't get so worked up or think that, oh my God, once we turn on the sympathetics in fight or flight, that means the heart rate's going to go through the roof and we're having these big fluctuations. No, we don't have very big fluctuations. They're actually quite mild fluctuations, but we can certainly increase the intensity of the nerve signals when we need to increase that intensity. And we can do it almost instantaneously. Think about if you were out driving and someone cuts you off, or a deer darts out in the road, and you didn't see it. Your heart races and you slam on that brake almost instantaneously because of your sympathetic nerves. Your sympathetic nerves will save your life because it helps you to respond now. The sympathetic nerves can become a chronic issue because if they're constantly wired and you're constantly stressed, your body can start to wear down over time. But having a healthy sympathetic response will, keep you going, keep you alive, keep your heart going, all that kind of stuff. Okay. If you want to learn, read about some interesting things, look up the word sympathetic dominance. Sympathetic dominance is a condition in which the sympathetics seem to predominate certain aspects of a person's physiology. And sometimes that can lead to a variety of like subclinical conditions or sometimes even clinical conditions. And believe it or not, you can actually treat sympathetic dominance using diet, nutrition, exercise, massage therapy, chiropractic care, biofeedback. There's a bunch of natural holistic ways that you can actually regulate sympathetic and parasympathetic function without having to be dependent on a medication to regulate their function. So just something to consider. Okay, but the chemoreceptors, the peripheral ones are located in the periphery. The central chemoreceptors are going to be located in the medulla oblongata, and it's the central chemoreceptors that are monitoring CO2 and pH. So it's just important to remember that. All right. So I briefly mentioned this other system. It is the Renin, Angiotensin, Aldosterone mechanism. It's one of my favorite mechanisms to kind of cover because it's one of those things that I think you saw this in when you discussed the Endocrine chapter, but it helps to tie it into what we're learning about. So the first thing you need to know is that the Renin, Angiotensin, Aldosterone mechanism, also known as the RAA mechanism, is going to be activated due to low blood pressure detected by the kidneys. So if the kidneys detect low blood pressure, the kidney will secrete renin. Okay? So low blood pressure secretes renin. So if I asked you, why does the kidney secrete renin? You would tell me in response to low blood pressure. So low blood pressure, renin is secreted. Renin then will convert inactive angiotensinogen into the active form angiotensin 1. And we're showing the liver here because the liver actually produces the protein angiotensinogen, and renin is what's needed to convert the inactive form to the active form. Angiotensin 1 is active, but it doesn't do a whole lot. It becomes more active when we convert it to angiotensin 2. In order to do that, we make our way into the lung capillaries. Once we're in the capillaries of the lungs, there's enzymes known as angiotensin converting enzymes that convert angiotensin 1 to angiotensin 2. This is also why, and I mentioned in the in-class Zoom meeting, I said that you may have heard of the term an ACE inhibitor. So what an ACE inhibitor does is it literally inhibits the conversion. So if I use an ACE inhibitor, what I'm going to do is I'm going to shut off the conversion of angiotensin 1 to angiotensin 2. So we won't make angiotensin 2. If I don't make angiotensin 2, that means I can't have the other effects that are going to occur. So that's what an ACE inhibitor does. Okay, I don't know what's happening there. Let me see if I can clear out my... Okay. All right, so angiotensin 2. Angiotensin 1 is converted to angiotensin 2, which is done by what's called the ACE, or angiotensin converting enzyme, and it's found in the lung capillaries. So angiotensin 2 is what does a whole lot of stuff. So angiotensin 2, even though it's not shown here, actually increases anti-diuretic hormone. It causes angiotensin, causes vasoconstriction, and you already know that vasoconstriction will increase blood pressure because when we shrink the diameter or we cause vasoconstriction, it increases resistance, which increases blood pressure. So vasoconstriction increases blood pressure. Angiotensin 2 also tells the adrenal cortex to secrete a substance, a hormone known as aldosterone. Aldosterone tells the kidney to reabsorb or hold on to more sodium. And when the body holds on to more sodium, it also holds on to more water. And so what that does is we increase sodium and water reabsorption, which decreases urine output, but it increases blood volume. Increasing blood volume also increases blood pressure. So, what are some things that that might be done? Like I said, I mentioned the ACE inhibitor would be a way to turn this system off. And you might be wondering, well, why would we want to turn off this mechanism? It's obviously being activated because of low blood pressure. So let me be more clear. It actually is measuring low blood pressure at the level of the kidney. If your sympathetics are chronically activated, so like chronic stress and other things that might cause a sympathetic dominant state, it actually would cause vasoconstriction, which limits blood getting into the kidney. When you limit blood getting into the kidney, it causes decreased blood pressure, right? Because we talked about vasoconstriction does increase blood pressure, but we're decreasing blood flow. And if I decrease blood flow getting into the kidney, that's why the kidney says, oh, I'm not getting enough blood. And so it's going to go through the mechanisms to try and increase the preload in order to increase the blood pressure to get more stuff in here. Now that's one situation. Also, if the kidneys are damaged or if the kidneys are failing, we'll also see an increase in run and release. And so an ACE inhibitor may be given as a way to decrease preload and to lower blood pressure. To my knowledge, if I may be speaking out of turn here, but I do believe that ACE inhibitors also are typically given in conjunction with like a beta blocker or a diuretic. I don't know that they're given in isolation, but they might be. So one of the other things that's released in response to the angiotensin 2 is ADH is released. So this is actually showing you the pituitary gland. Okay, so the pituitary gland. So I know sometimes it looks like the testes, but this is actually talking about, here's the neural stalk, and then it tells you it's the pituitary. That's a good way to remember it. Also, there's no other other structures that would indicate that this is the male anatomy. This is the pituitary, which is found in the brain. So there's a variety of things that are going to actually trigger ADH release. So you can see that osmolarity, meaning the concentration in the blood, will be associated with ADH release. In fact, if you have too much, if your blood volume is too low and or your solutes are too high in your blood, it'll increase thirst receptors and will increase ADH secretion. So, and what that does is it's going to cause vasoconstriction because we still want to cause blood pressure to increase. But it's also going to cause ADH specifically will increase water reabsorption while we're trying to consume more water. So the thirst reflex is not the same thing as ADH, but the same thing that triggers ADH is the same thing that triggers your thirst response, which is your basically how salty your blood is, just to leave that alone. You could also argue, well, wouldn't sugar do the same thing? Yes, sugar does exactly the same thing. Too much sugar causes you to retain water, and it also makes you excessively thirsty. And when you're drinking too much, then you start to void out excess, and then you have a whole host of problems. Vasopressin is also is the other term for anti-diuretic hormone. It's the more clinical term. Problems that could occur with ADH include diabetes insipidus. And there's two types of diabetes insipidus, or two main types of diabetes insipidus. You have central, also known as neurogenic, and then you have nephrogenic. Okay, so neurogenic, neuro referring to the brain, or central diabetes insipidus, DI, insipidus, and peripheral or nephron based diabetes insipidus. And so there's two types. If it's because of a central type of diabetes insipidus, basically the person is either not producing enough anti-diuretic hormone, or they're producing, or it's not being produced at all. Okay, so it's either producing too much or not enough. And what happens is, if you don't produce enough anti-diuretic hormone, it causes you to void more urine. You decrease your blood volume, but you're also increasing your urine output, which would make you thirsty. And so diabetes means more about your water intake and your water output than it does have anything to do with sugar. Diabetes mellitus is the one that you've heard that's associated with sugar, and diabetes mellitus is the one that has type 1 and type 2 diabetes, whereas diabetes insipidus has everything to do with the anti-diuretic hormone. The peripheral or the nephrogenic version of anti-diuretic hormone means that the ADH receptors are not responding to ADH. And so what happens is, you might have plenty of ADH, but when they get to the kidney, they're not doing anything, okay? Because the receptors are missing. The receptors are turned off, okay? So a lot of times people who have nephrogenic diabetes insipidus, it's a very, very problematic thing to have because it's difficult to treat. There are medications and there are things to treat it, but when it's neurogenic or central, where it's the pituitary is the problem, they can actually just take ADH, either as a nasal spray or it gets injected, and they can utilize that to help regulate their blood volume. As an additional side note, ADH inhalation has been used as a way to get people to study vast quantities of information in a short period of time. This is not a recommendation, but I think the application of that is interesting, and you can look that up. Just type in like ADH learning or ADH or vasopressin learning, and you can kind of see what shows up with that. And then I told you I like this graphic better than some of the other graphics, just because you can follow the flow chart, and I'll leave this up and I'll let you guys you can pause it at this point and just look. So what happens? So here's our blood pressure range. What happens if we go above our normal blood pressure range? And then you can see all the different mechanisms that become activated in order to bring it back down. What happens if it gets too low? Then you can see all the mechanisms we have to bring it back up. And so sometimes the mechanisms are turning something on or turning something off. Sometimes it involves the neural response. Sometimes it is an endocrine response. But oftentimes it's both neural and endocrine both working together to bring us back into homeostatic balance. And then I showed you this as well. This is just the baroreceptor reflex. I showed you this in chapter 19, I believe, in the heart. And it just talked about these things as far as regulating heart rate and stroke volume. But the previous slide, this one here, I think goes into this in better detail, and it covers some of the things that are covered in the next slide. So review this, read through these, and if you have questions, you can let me know. Okay, the last thing, this is also in your book, so you can see all the different factors and how they cause vasoconstriction, vasodilation. Remember, those are how we are regulating blood pressure. All right, the last two slides are on shock and edema. So there's four main types of shock. Hypovolemic shock, where there's a great loss of fluid volume. That could be due to hemorrhage. So think about like a severe car accident, or an arterial bleed, or after birthing a child, there might be excessive hemorrhage that could lead to extreme fluid loss. There could be severe vomiting and diarrhea. So in certain types of foodborne illnesses where you get excessive vomiting and diarrhea, that could lead to hypovolemic shock. Excessive burns, because you're basically losing a lot of fluid via the injuries that have been sustained. And then ketoacidosis can also lead to hypovolemic shock. One of the things I'll tell you to know is don't confuse this word ketoacidosis with the word ketosis. Ketoacidosis is a condition that typically affects type 1 diabetics, in which they are overproducing ketones and they have high blood sugar, and it causes detrimental and sometimes lethal effects within the body. Whereas ketosis is the release of ketones in a low blood sugar environment that can actually be, I don't want to say healthy and make that be like a controversial term, but it would be within the realms of normal variables when we're talking about ketosis, because we've all experienced ketosis at a point in our lives. When we're born, we eat, typically, if you're not being breastfed, you're probably on some sort of formula, but if you're being breastfed, that's pretty much just fat and water. And so as a newborn, you are in a constant state of ketosis. As you get older, and when you fast, meaning you go to bed and sleep throughout the night and wake up after you've been fasting for however many hours, you go into a mild state of ketosis. And so I don't want you to confuse the words ketoacidosis and ketosis, because they are two completely different words. Okay. Cardiogenic shock. This is now the heart it is unable to maintain cardiac output. So cardiogenic shock would occur if somebody went into cardiac arrest, or if someone went into cardiac or had a myocardial infarction. This will just be like a little bit of like a bonus activity, just to see if you have been listening to this. So if you're listening to this, what I want you to do is send me an email and just briefly describe the difference between cardiac arrest and myocardial infarction. So what is the difference between cardiac arrest and heart attack? Okay. And I'll even help you out. If you go to the American Heart Association, if you go to heart.org is what I believe it is, they have an infographic that easily defines the differences between cardiac arrest and heart attack. Okay, so make sure you can look at that. Email me, and I will give you a few bonus points for doing that. Vascular shock is anything that's going to affect vascular tone. And with vascular shock, typically what's happening is you get dilation. So vascular shock is also known as vasodilatory shock. And it's also known as, what's it called? Oh, there's another term, and it's completely, I've lost it. I'm sorry. So, is it just dilatory shock? It might just be dilatory shock. I know it starts with a D, but I felt like it was a different word. So, but I'm sorry, it's distributive shock. So another term for vascular shock is distributive shock. And so distributive shock includes septic shock, anaphylactic shock. It includes nervous shock. So like if somebody has like a spinal injury, that will lead to distributive shock, or can lead to distributive shock. Septic shock is typically due to a bacterial infection, and it causes vasodilation in the blood vessels. And we'll talk more about this and anaphylactic shock when we get into chapter 21 on the immune system. Anaphylactic shock can be due to like peanut allergies, a bee sting, or anything, or shellfish allergies that could trigger a response in which the respiratory bronchioles constrict, so you can't breathe, and then you get rapid vasodilation, which increases swelling, which again means you can't breathe. And then that's why with anaphylactic shock, they'll give someone an EpiPen to cause vasoconstriction and bronchial dilation. And again, we'll talk more about that in the next chapter. Obstructive shock means that there is some sort of obstruction in the vascular system. So it usually means like an embolism or a thrombus. So a clot has dislodged and made its way throughout the body. But it could also mean tamponade, which means that there's fluid buildup around the heart. And what it's doing is it's compressing the chambers of the heart, and so that they can't beat appropriately. And so it decreases cardiac output. If you want, and I recommend, go on to YouTube and just type in cardiac tamponade, and they have some really good videos showing what cardiac tamponade is and how it affects blood flow. The last slide here is on edema. So we talked about how fluid gets moved into and out of the tissues. And edema means that we have too much fluid in the tissues and it's not being reabsorbed, or it's being pushed in faster than it can be reabsorbed. So there's a number of causes that can trigger edema. There could be kidney failure. So think about it. What do the kidneys do? The kidneys either help you retain water or get rid of excess water. So if your kidneys are failing and they're failing to get rid of excess water, that water's got to go somewhere. It's going to go into the tissue spaces. There's a term for this. It's called third spacing. So basically the third space is within the tissue spaces. Histamine release, particularly for when there's excess histamine release, will cause edema and swelling. Some of that histamine release is expected to occur in the event of a trauma or an injury. You are going to have edema as part of your normal inflammatory response. Liver disease. If you remember, we talked about colloidal osmotic pressure, and I said that if the liver is diseased or failing, it will not produce proteins, and those proteins are needed in order to contribute to the colloidal osmotic pressure. And so if we don't have the proteins in the blood, then the blood can't hold on to its water, and it makes its way into the tissues. And then lymphatic obstruction. The best way I can tell you that's helpful for this, if you've ever known anybody that's had a lymphadenectomy, that usually follows like a mastectomy. So like a woman who's had breast cancer or is at risk of breast cancer and she gets a mastectomy, or if she had breast cancer and they had to remove the breast tissue and the lymphatic tissue, she's at greater risk of developing fluid retention like in the arms because they're not being drained appropriately. We'll talk more about lymphatic drainage next chapter. So what are some consequences of tissue edema? Well, it can lead to necrosis, which means that you have dead tissue in the area. It could lead to pulmonary edema, which is fluid that starts to build up around the lungs, and it makes it more difficult to breathe. And we'll talk about pulmonary edema when we get to the respiratory chapter. You can get swelling around the brain, which is going to affect your cognitive responses, and it may even lead to coma or death, which can then severe edema or circulatory shock also could lead to death as well. When there's excess fluid in the tissue spaces, it leads to low blood volume and low blood pressure, which means we're going to have significant changes in our fluid or in our perfusion rates, which will also be devastating. So if you have questions, let me know. Don't forget to email me that bonus opportunity. And if you don't know what I'm talking about, rewatch the video and you'll find it where I said it clearly. And if you have questions, please let me know. All right.